Cellular neuroanatomy is another topic that a lot of students have difficulty with. It is a big, complicated topic. In all of the PowerPoints during this review session, I might be using words or terminology in a different manner than it was used the first time you were taught. That's okay. I have been accused sometimes of speaking beneath my students. You know what? That's okay too. I mean, it's always good to hear a lecture and say to yourself, wow, that was easy. You know, that's good. It means you really understand everything. But if I say something a little bit differently than a previous teacher, it doesn't mean that teacher was wrong or I'm wrong. It's just we have different ways of explaining things. Embryonic development. Well, I think everybody knows that when an egg and a sperm unite, we get an embryo. The very first thing that develops is the central neural plate, so it's kind of flat. Then the edge of that plate is going to start to fold and join together, and it's going to form a neural crest on the outside edge of the plate. That crest is going to fold together and join, and it forms a tube-like structure. That's the neurotube. It's eventually going to be our spinal cord. Improper closure of that tube could lead to spinal bifida. So here's a picture of a sperm on the surface of an egg. This is not necessarily something we're going to be tested on, but I just thought it looked really cool. This is a 6-day embryo on the wall of a womb. Again, thought it was really cool. Okay. So neurons, they're one of the very first things that develop when we're an embryo. Neurons are the primary component of the central nervous system. Neurons are what are responsible for the EEG activity that we're recording from. They are incapable of reproducing themselves. So if we have a loss of oxygen, anoxia, if we damage those neurons in any way, that patient's going to have permanent brain damage because we can't replace our neurons. Neurons are the bridges between the central nervous system and the peripheral nervous system. They allow the two systems to communicate with each other. Therefore, a neuron can send information and receive information. Very important. On the right, we have my lovely artist rendition of a neuron. A lot of times in my slides, I try to use real pictures if I know for a fact that, my school or whatever has permission to use that picture, or I'm allowed to use a picture from a textbook. But sometimes, I don't want to be accused of plagiarism, so you'll see some of my little cartoon-ish drawings. Okay, on the left we have some definitions of the parts of a cell. The cell body. Well, that's what I call it, but you might be reading a textbook or maybe on a board exam and they're going to call it something different. It is also called the soma and the perikaryon, and I won't make you think that I know how to pronounce that word. I've never seen it referred to as that, but again, anytime there is more than one way to say something, I always try to give you every name for it because you never know what they're going to call it on a board exam, and I don't want you to get confused. So make sure you know that all three of those things mean the same thing. Okay, so the job of the cell body is to direct metabolic activity. It's going to decide if messages that are sent to it are important or not. The cell body has many dendrites that come out of it, and the dendrites receive information, and a dendrite is the part of the cell that is creating the EEG activity. And I underline the R in dendrites and the R in receive because rights receive. There's usually one axon and it sends information away. So I underline the two A's for axons away to help you remember which one is which. There is a synapse, and that is something that can happen, whereas the cleft, the synaptic cleft, is a place. So the axon of one cell, and we see this in our picture here in the top right corner, we see the axon of a cell that's off the screen, and it is coming in close to the dendrite of the cell on our screen. It is not touching it, it just got very, very close. That space in between is the synaptic cleft. That axon is now going to dump some neurotransmitter substances into that cleft, and the dendrite is going to receive it and send the information to the cell body. And remember, the cell body's job is to direct metabolic activity. Metabolic means chemicals. So it's going to decide, is this information that was just sent to me from another axon, is that important? And if so, I need to send it down my axon to the dendrite of another cell and send this information on. Myelin, it surrounds the axon and it increases the speed of how fast these signals can travel. Myelin is a white milky substance, but in my picture here it's been shaded in as yellow. The nodes are something that allow the signal to jump and therefore move faster because it can jump from one node to the next. Teledendra are fine little branches on the very end of an axon. Okay, so you looked at my cartoon version of the cell. These are real neuron cells under a microscope, and even though this has been magnified really, really high, you still can't really tell the difference between what are the axons and what are the dendrites because there's so many of them and they're so close together. Neurons are tightly packed together so that when you and I look at the brain, you and I can't see individual cells. We have to put it under a microscope to be able to see the individual cells. So sometimes these cartoon pictures help us see things a little better. Here's a cartoon picture of a synapse. So we have the axon of one cell, and it's coming close to the dendrite of another cell. The axon has a presynaptic terminal that have little vesicles in them. And just think of these vesicles as jars. And they contain chemicals. They contain neurotransmitter substances. When they pull in to the synaptic cleft and they get close to that dendrite, they're going to open up and dump the neurotransmitter substance into that cleft. Then the postsynaptic terminals, which are vesicles within the dendrite, are going to receive that information and then send it to the cell body. Now, the picture that I showed you of the neuron that we memorize in our field where it has one axon and several dendrites, actually there are several different kinds of cell structures. So that's not the only one, and your textbook might show you several different cell structures. So I don't want you to think, gosh, she doesn't know that there's more than one type of structure. I do. But the one that I'm showing you in this PowerPoint today is the cell structure that they want us to memorize for the board exams in our field, whether it's EEG or evoked potential or whatever. So with what I described, we're going to have the axon of one cell communicate with the dendrite of another. So that's on the bottom of this slide. Axodendritic. That is the most common form of communication between neurons. However, remember there's other kinds of neuron cells, and some of them are going to communicate from one axon to the soma or cell body of another cell, or it could go axon to axon or dendrite to dendrite. So I just want you to know there's more than one way for neurons to communicate, but the basic structure that we learn is the axon of one cell communicates with the dendrite of another. There could be a board exam question that says which of the following is the most common way for neurons to communicate. We have lots of different cells called receptor cells. They're all over our body, they're on the inside, they're on the outside. We have receptor cells all over our skin so that the minute we touch something or something touches us, that receptor cell is going to receive that information and send it up to the brain for us to interpret what just touched us. Was it hot? Was it cold? So receptor cells are responsive to and can discriminate between different stimuli like light, sound, heat, cold, pain, pressure, change in position, you name it. Effector cells are cells that contract and relax muscles, or They can secrete hormones or other chemical substances like what glands do, and they enable the organism to make a specific adaptive response to that stimulus. An exteroreceptor is a sense organ that responds to a stimulus external to the body, such as touch, pressure, temperature, pain, whereas an interoceptor is a sense organ that responds to stimuli internally, so within the body. Proprioceptors are sense organs sensitive to changes in position of the body, joints and limbs, vibration, shape, form, weight. They're mostly in the parietal lobe. That's where they're interpreted at. So I always remember P for proprioceptors. They tell us about position, which is P, and they're interpreted in the parietal lobe, which is a P. So those are easy for me to remember. Now, these are not all the kinds of receptor cells. There's tons of them. There's chemoreceptors for taste and smell, thermoreceptors that detect changes in temperature. There's nociceptors, which are pain detectors, and on and on and on. But I wanted to give you an idea of another kind of cell. The third kind of cell that we're going to talk about are neuroglia. The word glia comes from glue because they glue the neurons together. So remember, we can't really see a neuron just by looking at the brain. We have to put it under a microscope. The glias are non-conducting. We said that a neuron can send and receive information. A glia cannot. And a glia can rapidly reproduce themselves. But we said earlier, a neuron cannot. If we lose our neurons, we're going to have brain damage, and it's probably going to be permanent because we can't get that back. But a glia, if it gets damaged, can rapidly reproduce itself. In fact, sometimes they reproduce too fast, and that's what causes brain tumors. All of your brain tumors are from the glia cells because they've over-reproduced and they've created a mass. So if you notice, your brain tumors are called astrocytomas or oligodendrocytomas and so forth. Okay, so we have four main kinds of glia. The top three we talk about a lot. The bottom one we just talk about a little when we're talking about the ventricles. An astrocyte provides support and nutrition and insulation for the brain. It provides structure and nutrition and insulation. An oligodendrocyte plays an important role in the formation and maintenance of myelin within the central nervous system. Microglia defend the nervous system against infection and injury. They can also transport waste away. Now, that ependyma, they line the ventricles and the central canal of the spinal cord, and they secrete cerebral spinal fluid. So these are all very important. Make sure you know them. Make sure you know what their function are. Ions. They're chemicals in the brain. We have chlorine, sometimes referred to as chloride. Make sure you know the abbreviation and whether these are negative or positive. So chlorine or chloride is negative. Sodium, Na, and it's positive. Potassium, a K, and it's positive. Calcium, Ca, and it's positive. Make sure you know these abbreviations and whether they're negative or positive. All right, so we've got the cell body, and it's resting. It's just sitting there. Nobody's bothering it. The neuron is quiet. The potential across the membrane, so that cell body that has chemicals or ions in it, is somewhere between -60 and -80 millivolts. Every textbook is different. Some textbooks say 70, some say 80, but make sure you know it's millivolt and not microvolt. Inside of the cell body, there are lots of anions, and we talked earlier about that's an ion that's negatively charged, so it's gained an electron. And there's also a little bit of potassium inside the cell. On the outside of the cell, in the extracellular fluid, there is a lot of sodium, which is positive, and a little bit of chlorine or chloride, which is negative. Oh, I had to have a beautiful artistic rendition of this. So intracellular fluid inside that circle there, that's inside the cell body. Extracellular fluid is on the outside of our cell, and we have a membrane that's in between that keeps them from going back and forth. The orange are big negatively charged ions, otherwise known as anions. So even though there's a little bit of potassium inside, there's more anions than there are potassium, so it's negative on the inside. It's negative 80 millivolts. On the outside, we do have a little bit of chlorine or chloride, which is negative, but we have more sodium than anything else, and that's positive. It's the difference between the inside and the outside, the fact that the inside's negative, the outside's positive, that difference is what maintains the resting membrane potential. Some definitions. The word threshold. It is the smallest degree of stimulation necessary to elicit an action potential. So if you're sitting in the classroom and there's a person in front of you and you're trying to get their attention without the teacher noticing, you might whisper and go psst. But the first time you do it, it's so soft that though you heard it, the person in front of you didn't. So you did not create an action potential. You did not reach threshold. So you're going to do it again, this time a little louder. Psst. Ah, this time they heard you. It started an action potential. So you were able to reach threshold. It's the smallest amount of stimulation where you can start an action potential. And it doesn't have to be sound. It could be touch, temperature, anything. Action potential. That's disturbance of this resting membrane that we've been talking about by some kind of a stimulus. And again, a stimulus, it could be chemical, electrical, temperature, mechanical, hearing, sight. Action potential. So we stimulated something, we reached threshold, and we depolarized the cell. Polarized means to either be negative or positive. To depolarize something is to make it less polarized. We'll talk about that more in just a little bit. A chemical substance is then released, and then calcium is released. Now, to say, well, a chemical substance is released is very vague. Well, because we're not studying to be, neuroscientists. We're studying to be technologists, and if I were to write down the real steps of an action potential, there's probably, 50 or 100, steps, and they're very long and they're very complicated. And we're just trying to simplify it so that we can understand it. So some chemical transmitter substance is released, and when that happens, calcium is then released. Calcium has a very important job. It opens the lid on the vesicle in that axon that contains the neurotransmitter substance. This is a very common board exam question. What is the role of calcium in an action potential? Its job is to open the vesicle so that the neurotransmitter substance can then be released. If the substance that was released is excitatory, sodium, which from my picture earlier is on the outside of the cell, it rushes into the cell, and that resting membrane is going to go from negative 80 millivolts to around positive 10 millivolts. So it went from negative to positive. It was depolarized. It was negative, but we made it less negative. We made it more positive. Now you might say, why did they say less negative and more positive? Well, because maybe it went from negative 80 to negative 1. That's the same difference. We still made it less negative or more positive, but we went from negative 80 to negative 1. Well, that's still negative. So that's why we like to say more positive or less positive instead of actually giving a number. Okay, so all the sodium rushed in. It went from about negative 80 to about positive 10. Now a little bit of the potassium is going to leave the cell. Okay, so I said to be polarized means to either be negative or positive. In an excitatory postsynaptic potential, we depolarize the cell, so we make it less polarized. So we took this cell and we made it less negative and or more positive, whichever wording you want to use. We went from that -80 to about +10. We increased the cell permeability to sodium. So that sodium, which was positive, rushed in. So that's why it went from negative to positive. The membrane potential falls. So in other words, when it's excitatory and we stimulate it, that membrane no longer keeps the sodium out and allows it to rush in, making it go from -80 to +10. Now, this cell is more likely to fire, meaning it's more likely to send a signal on to the next cell. And it says, oh, this information is important. I must tell someone else. So remember, the cell body or the soma, its job is to direct metabolic activity. Metabolic is chemicals. So when the cell body receives these chemicals, it came from the axon of another cell to the dendrite of the current cell. The dendrite sent it to the cell body, and now the cell body has to decide, is this important? And if so, it's going to be more likely to fire and send a signal on to the next neuron. So again, we have our resting membrane potential. It has a membrane around it that's keeping the sodium out. But if we stimulate it, and if we reach an action potential, so we reach threshold, and a neurotransmitter substance is dumped out and collected, and if it turns out that it happens to be excitatory, then that cell might send that information on. Inhibitory postsynaptic potential. Instead of depolarize, we hyperpolarized. We make the cell even more negative than it already was, because in an inhibitory, we increase the permeability of chlorine from the outside to sneak inside that membrane. It's going to stabilize the membrane instead of making it fall, and it's going to be less likely to fire. So the cell body is going to say, you know what, I really don't think that this is all that important, so I'm not going to send this signal on. I used to always ask, well, why on earth would we want an inhibitory neurotransmitter substance? Why would we not want this information to travel? Well, we use inhibitory chemicals when the brain seizes. So if you've ever seen on an EEG, we get a spike and then we get a wave, and a spike and a wave during a seizure. Well, the spike is the brain being bad. The brain is hyper-excitable. It sends an excitatory substance when it's not supposed to, and it's firing and it's not supposed to. Well, the brain knows that. It goes, oh no, I shouldn't have done that. And so an inhibitory signal is then sent out to try to stop it or calm it down. And that's the wave that we see after the spike. It's the actual brain trying to correct itself. And sometimes it has to send so much of that inhibitory substance. You'll notice that our EEG during a seizure will go from all this spike and wave, and all of a sudden the seizure part is over, the spikes have stopped, but the patient's EEG record is very slow because it sent so much of the inhibitory. So it might seem weird. Why do we have inhibitory substances? Well, it's to help the body kind of counteract itself. We don't always want to fire. So depolarized was to take that cell that was already negative and take it to the opposite direction, so less negative or more positive. Hyperpolarized is to take it's already negative, make it more negative, so it's hyper, it's over the top, or it's already negative and we're going to make it more negative, so it's hyperpolarized. Now, after an excitatory postsynaptic potential, we have a refractory period, and there are two of them. An absolute refractory period is when the cell is incapable of responding to another stimulus. This is why sometimes during a nerve conduction or evoked potential, we cannot stimulate boom, 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 boom one time after another. we have to wait a little bit, like at least a few milliseconds, to give that cell time to reorganize or repolarize and get back to resting again. If we were to stimulate during a nerve conduction or evoke potential too quickly, we're not going to get a waveform on the screen. It's going to be flat because it's in an absolute refractory period and the cell is incapable of responding to the stimulus that we gave it. If we wait just a few split-second, microseconds, we go from this absolute refractory period to a relative refractory period. This means, well, I could stimulate the cell at this point and I will get a response, but my response on the screen is going to be less amplitude than if I would just wait a little bit more, and it's going to take more to reach threshold. So let's say that for a patient, their hearing threshold was 15 decibels, but they're in a relative refractory period. So to get a response on my screen, I might have to stimulate 30 decibels to get the same kind of response. Okay, so we've gone through our relative refractory period, and now the cell has to repolarize itself. It has to get back to resting again so that it can start to receive more stimuli. We have something called a sodium pump, and it forces the sodium out of that cell, kind of like a sump pump that's pumping out water from your basement. It is also known as the active transport pump, ATP. And ATP happens to be a neurotransmitter substance that contains a stored energy that is released to help repolarize the membrane after an action potential. So we're going to look at ATP more in a minute when we look at a list of neurotransmitter substances. But that's the role of this particular substance, is to help get that sodium out of the cell and get it back to resting again. So we have lots and lots of neurotransmitter substances. They always make us memorize, though, 3 of the main excitatory substances and 3 of the main inhibitory substances. Well, acetylcholine, or ACTH, is on both lists, so if you can name that one, you're good to go. Then the other 2 excitatory that they always want to make sure we know is L-glutamic acid and L-aspartic acid. So to remember, oh gosh, is that the excitatory one or is that the inhibitory one? I want you to picture this cat, and I want you to picture that acid has just been thrown on him. If acid had been thrown on you, you'd be pretty excited. You'd be screaming, your eyes would be big, just like this cat, and you'd be trying to get it off of you. So think of acid being thrown on you as an excitatory transmitter substance. Here are our inhibitory neurotransmitter substances. Again, we've got that acetylcholine, so it's going to work in both columns. And our 2 substances now are GABA and glycine. They both begin with a G. So think of a gentle giraffe. It's inhibitory, not excitatory like acid. Here are some other substances. Granted, is it all of them? Lord, no. There's tons of them. They're probably discovering more and more all the time. But these are some common substances, neurotransmitter substances, that you should try to be aware of and be able to recognize. And ATP is on the list there. That's that one that helps repolarize the cell and pump all that sodium out. I was talking to you earlier in the PowerPoint about how there are actually lots of different kinds of neuron cells, and they're all shaped differently, but we tend to memorize that same structure of one cell body, one axon, several dendrites. That's what we tend to learn for our board exams. But I realize, you've got textbooks and maybe other handouts that talk about other types of cell structures. So here's a picture of just some neurons and some neuroglia. Again, nothing you have to memorize or label or anything like that, but just to kind of get an idea of the different cell structures. All right, that's going to do it for cells. I hope that you found that as exciting as this cat and this baby find that fish to be.